Smoky mice are interesting for many reasons. They're cute, they're fuzzy, they smell nice actually, but that's not at all the reasons why I'm interested in them. I'm really interested because they're enigmatic and they're part of an amazing diversification of species that occurred in Australia once rodents arrived 4 million years ago. They're also a member of a group heavily impacted by extinction since the arrival of Europeans, with more than 40% of all mammalian extinctions in Australia being rodents. Smoky mice are one of the survivors, but they're also an endangered species threatened with extinction. The traditional way that we collect samples without sacrificing the animal is we take a small ear biopsy, a little sliver from the edge of their ear, typically stored in some sort of preservative that preserves the DNA. Because we're trying to preserve the genetic diversity that's living in populations today, we now use techniques that preserve the living cells, and then we've been able to grow them in our facility at Melbourne Museum. That little bit of ear contains skin cells, and we bring that back and take a little piece of that and score it into a petri dish with a cell culture medium, a medium that those cells love to eat with all the food and nutrients they need to grow. And then over a matter of a week to 4 weeks, they grow in a low oxygen incubator. So once we've grown the cells to sufficient quantities, many millions of cells, we can sample them and then store them. We put them into one of our cryotubes and then slow cool them down to liquid nitrogen temperature. That's minus 196 degrees Celsius. At that temperature, they're basically cryopreserved alive in stasis. And we can pull them out at a later date and thaw them out and then grow those cells again. We can do studies on those cells, or we can use those cells to modify and reuse in the wild.
Museums Victoria respectfully acknowledges the traditional owners of the lands where we work, the Wurundjeri and Boonwurrung peoples of the Eastern Kulin Nation, and First Peoples language groups and communities across Victoria and Australia. We pay our respects to their elders past, present, and their emerging leaders. Our organisation, in partnership with the First Peoples of Victoria, is working to place First Peoples living cultures and histories at the core of our practice.
Welcome everyone. My name is Kate Phillips and I'm the Senior Curator of Science Exhibitions at Museums Victoria. It's my great pleasure to welcome you to Melbourne Museum tonight for this special event, and a warm welcome to those joining us online as well. I begin by acknowledging the Wurundjeri Woiwurrung and the Boonwurrung peoples of the Eastern Kulin Nations, the traditional owners of the land on which we work and where we gather this evening, and I pay my respects to ancestors and to all elders and First Nations people with us this evening. I'd also like to thank Cindy and Gab, who are providing our Auslan interpretation for tonight's program. Tonight's event is part of Future Forums, Ideas Shaping Tomorrow, a series of monthly conversations proudly presented by Museums Victoria that brings together the world's brightest innovators and creative thinkers to reflect on the big ideas and important questions that shape our future. Today's program, The Genetic Rescue of Our Fantastic Beasts, is presented in association with the International Congress of Genetics being held in Melbourne this year, and welcome to any of you who have come from the conference. And that conference tonight brings together experts in conservation genetics from Australia and overseas to examine the latest breakthroughs in genetic rescue. It comes as a timely moment as just a few weeks ago, a species that has not been seen for more than 50 years and was feared extinct in the wild has been rediscovered, and we'll be talking more about that tonight. We're delighted to be hosting this event. For over 165 years, Museums Victoria has been collecting, identifying and recording the biodiversity of this region. And as you saw in the films, using latest techniques to help do that, our collections contribute to a baseline for measuring environmental change and biodiversity loss since European settlement. And we're supporting conservation research and driving groundbreaking projects to protect critically endangered species and ecosystems. At the Museums Victoria Research Institute, we're undertaking world-class wildlife genomics research to inform conservation strategies for threatened species. Working in collaboration with government organisations, universities and community partners across Australia and internationally, the museum's research plays a critical role in understanding and protecting our biodiversity. Now it's my great pleasure to introduce our host for the night, ABC Radio National's Natasha Mitchell, to introduce our guest speakers this evening. Thank you, Natasha.
Thank you so much, Kate, for the wonderful warm welcome. Hello, welcome. How are we tonight? Excellent. How are we tonight? Excellent. Great to have you here. I'm especially excited to be hosting this event to coincide with the International Congress of Genetics. The geneticists are in Melbourne right now. Who is a geneticist? Yes, nice. You can say that. You can really throw your hand up with pride in this crowd. It's wonderful to have you here. If you're joining us from overseas, welcome to Australia. Welcome to wonderful, balmy Melbourne, midwinter. It's great to have you here. And I'm really looking forward to having you post your questions to Slido. Now, did I have the instructions? Yes, I did. Have I put them somewhere ridiculous? Yes, I have. You need to go to slido.com. So get out your phones. Just pop your phones on silent tonight, if possible. That'd be great. And the code is RESCUE 1907. Pop that into. So slido.com, rescue 1907. I'm going to weave your questions in throughout the event once we get kicked off. Also, just to let you know that we are recording tonight for the show I host at the ABC, Big Ideas, which is our live event show. You can find it on the ABC Listen app as a podcast, and it goes out on ABC Radio National as well. So tonight we are on a rescue mission to save fantastic beasts, a genetic rescue mission, and we're going to find out what that actually looks like. I want to acknowledge that tens of thousands of years of knowledge developed and held by First Nations people about the lives and the needs of the species that we share the planet with. We are building on that knowledge that has occurred over tens of thousands of years. So where else in the solar system do we find the vast number of species that call this singular, precious planet home? Nowhere. What an extraordinary thing that we are the planet full of rich, diverse life. Every time I think about that, I'm just blown away. So the fact that so many of the Earth's fantastic beasts are really facing a perilous future right now is completely shocking to me. We are concreting and we are cutting down and we are clearing all the places and spaces that all those wild animals call home, all those fantastic beasts. So, and we also have climate change, which is changing their future in the most extraordinary and difficult and complex ways. We are part of the problem, but are we part of the solution? And wonderfully, tonight is a story of hope and possibility, because we are very much a part of the solution, and genetic science could also very much be part of the solution. So we have three fantastic speakers, incredible women. Our special guest from Kenya is Professor Anne Muigai, who is a molecular population geneticist. She is Deputy Vice-Chancellor at National Defence University in Kenya. Her research focuses on the conservation and climate change resilience of not wild animals, but of indigenous African farm animals, sheep, cows, goats, and we're going to find out why that is so vital in Africa. She's also chairperson of the African Biogenome Project, which is an immensely ambitious project to sequence the genomes of, is it 100,000 animals and plants?
100,000 in 10 years.
Amazing, and they need all the money they can get to do that. We've got Dr. Joanna Sumner, a conservation geneticist right here at Museums Victoria. She's Senior Manager of Genetic Resources, which means she manages the museum's wildlife DNA collection, which we're going to find out more about. She manages the Ian Potter Australian Wildlife Biobank and facilitates research in the museum's DNA laboratory, and she has a slight obsession for reptiles, I gather.
True.
Just a little bit.
Won't deny it.
A little bit of obsession. We have Dr. Marissa Parrott, welcome, reproductive biologist with Zoos Victoria. Her research is all about improving the success of captive breeding programs and also the process of releasing those bred species back into the wild and so that they may thrive and survive. So marsupials and rodents and froggies. Zoos Victoria has this ambitious goal you might have heard about, that is, on their watch, no Victorian terrestrial vertebrate species will go extinct. That's the ambition. Let's give them a big whopping welcome. So I want to start with a question for all of you, that is, when you wake up in the morning, why do you do the work you do? Why are you so focused on saving species?
I think it's similar to what you said in your introduction, is that when I think about the evolutionary history of the world around us and that the species that we see have been here for millions of years, happily breeding and surviving and changing gradually, that I can't bear the thought that that has been destroyed in a few hundred years. It feels like an extraordinary waste. So I think for me, it's about wanting that history to continue. I love the biodiversity and think of, when I think of it, I just want it to continue.
That deep, deep, deep history. Well, we're only just a pinprick in time, aren't we?
Absolutely, yeah.
Yeah, that's Joanna's answer. What about you, Anne?
Well, in Africa, animals have been an integral part of our lives. And just walking or driving from one place to another, you come across a herd of zebras. And over the years, we're seeing that declining, and through climate change and expansive agriculture. And just, watching the losses happening before our eyes is a call to action. So wanting to make a difference and wanting to conserve this, because this is a natural part of our heritage, and we have a duty to make sure it exists for the future generations to come.
It is extraordinary, isn't it, just in our own adult lifetimes that we witness these radical changes in what species are around us.
True, and just even remembering from my childhood, driving through one village to maybe the city, the change in the environment has also, the trees are gone or going, the landscapes are changing, and that has a tremendous influence on the livestock and the plants and the species that actually live there. And also the activity of man generally, who have come with plants that are not indigenous to Africa, and the implications and some of the damage it has done to the environment in our own time.
Yeah. Marissa Parrott, what about you? What gets you up in the morning?
I've always loved wildlife. Ever since I was a tiny girl, I loved wildlife and I wanted to help them. And so now, looking around the world, it breaks my heart that we're in a global biodiversity and extinction crisis. Wildlife populations have declined by about 69% on average across the world, and Australia is leading the charge, and not in a good way. In the last 200 years, we've lost more mammal species than any other country in the world. We currently have 1,900 species and ecosystems threatened with extinction.
1,900. Did you know that? I didn't know it was that number.
It's horrifying. And I get to work with these animals. I get to work with the tiny frogs and the little mountain pygmy possums and the tiny smoky mice that we saw just before on the screen as well. And I love them. And I can't imagine a world that doesn't have them. And so every day I get up to make a difference, as Anne said, to fight extinction, to get these guys back out in the wild where they belong and safe So hopefully in the future they don't need us anymore and we can move on to the next sort of species that do.
You have such a cool job as an endangered species reproductive biologist. I think of you as a kind of combination of a sex therapist and an IVF doctor for wild animals. Is that right? So you're kind of getting them together. Come on, mate, get it on, and then you help them breed, you know.
Absolutely. It's all about setting up the right scene for our beautiful animals. It's about getting them comfortable to have those relationships and then have those healthy babies that we can get back out in the wild so that we've got them out there. So it's really about having those strong breeding programs, having good genetics and saving those species. Little Barry White probably doesn't hurt either.
Yeah, what does the mountain pygmy possum like?
Sexy partners who smell different to themselves. So we've done the mate choice on our beautiful mountain pygmy possums, and they like genetically dissimilar animals. So for them, it's not about the sound, it's all about the smell.
The smell. Now, there's a very special critter that has been in the headlines, and so much so that federal politicians, state politicians took to the stage when it was announced. Why? Why did all the political heavyweights take to the stage just recently?
So
Joanna Sumner.
This was a very exciting find. So in January of this year, there were some ecologists who were out doing some surveys west of Melbourne there was a potential development sites, and they were looking for one particular lizard, a legless lizard, so they had some tile grids out, and which are just roof tiles, and if you leave them on the ground, then reptiles like to sit under them because they're nice warm spots.
Is a legless lizard basically a snake?
No, it's a skink without legs.
Of course.
Mostly.
Right.
Except for the ones that are geckos without legs. So it's something that's evolved. Leglessness has evolved many, many times in reptiles. But these ecologists came across a dragon. So dragons are quite, they always have sort of spiky skin, and this one definitely had legs. And it was clearly not the legless lizard that they were looking for.
But it didn't have ears.
It didn't have ears. So they realised quite quickly that this was potentially the Victorian grassland earless dragon, which hadn't been seen since 1969 and was presumed extinct. So it hadn't been declared extinct, but it was more than 50 years since it had last been seen. So it was getting very close to that time. So and it did in fact turn out to be this species.
So just give us a sense though of that moment, because this Victorian grassland earless dragon had been an elusive enigma, hadn't it? It had totally captivated the scientific community. Why?
Yeah, decades of work and thousands of hours of research and fieldwork had been undertaken specifically looking for this species. So they're quite little lizards. They're probably only about the size of your hand at the most. So, in fact, yes, a small hand. And they live inside spider burrows. So they're not something that sits out on rocks very often. They're really quite cryptic. So they're small. You don't see them very often, and it's often very hard to know if they're in a habitat. So they also live in grasslands, which is now, I think, the grassland used to cover a major proportion of Southeast Australia, and I think it's something like 99.5% have gone.
So less than 1% of grasslands in Victoria or Australia or Victoria
in Victoria
have gone.
Yeah, have gone.
So this species though, what was significant here is it looked like this would be the first lizard species to go extinct on mainland Australia. So joining the 31, I think, reptiles globally that have gone extinct.
Yes,
so this was a really significant loss.
Absolutely. And this species had it was thought up until about 4 years ago that this was part of a broader group, or that the species was broadly distributed through Southeast Australia, but it was actually split into 4 species. And so this one has a very, very narrow distribution, really only just in the west of Melbourne. But with the discovery of this species, one of the things that we then had available to us were all the DNA technologies that we are learning about and developing in looking at species. So one of those is eDNA. So this is environmental DNA. So now that we have DNA from this species, which we didn't before, because in the 60s we a lot of the technologies weren't available then, we can now look for the DNA of this species in the environment. So swab around spider burrows and see whether we can find the DNA in the environment. And that's one of the projects that Museums Victoria has been developing with Zoos Victoria in helping to conserve this species now that we know where it is. Well, one small area.
So, Joanna, Marissa, what was unleashed when that? How many individuals were found, and how significant was that from where you're sitting?
This is amazingly significant. It's so fabulous to find a species that you thought was lost for 54 years. And at Zoos Victoria, they're one of our fighting extinction species that are on our list for prioritisation. We've been actively searching for them for 10 years. We always had the hope that they were out there. And we're actually working with a similar species called the Canberra grassland earless dragon, where we've been developing our husbandry and our enclosures. We've bred them to help Canberra. We sent 136 young dragons back just last year. But it means that we had everything ready to move when this exciting discovery happened. And so we now have 16 Victorian grassland earless dragons with us safely at Melbourne Zoo. We're working on the genetic diversity of the species, biobanking with Joanna and with Museums Victoria, and we've started a new conservation breeding and insurance program to build up their numbers, to then get them back out safe in the wild, and to start new populations where we can keep them safe as well, where we can find those really important pockets of grasslands. And to look for other populations too, using the eDNA and even our own detection dog team, so that we can go out, we can find these animals, we can rescue the animals and their genetics, and we can keep them safe so that we're not going to lose them.
Oh, we must come back to that because, you're putting them back out into an environment where they're vulnerable and that is under threat. But can you just describe then what genetic rescue means for the grasslands earless dragon without the ears? I can't believe a lizard doesn't have ears. But anyway, go on.
Certainly doesn't need them, so it's okay. Genetic rescue is needed because many of our populations are small and they're isolated and they're fragmented. And so over time with these small populations, whether it's grassland earless dragons or mountain pygmy possums, those numbers start to decline and their genetic diversity starts to decline. And the genetics is the building blocks of life. So if you don't have healthy genetics, you don't have fitness, you don't have good breeding success, you don't have resilience for the future so that you can respond properly to things like a changing climate. And so through genetic rescue, we can do a number of things, and that's look at the different populations, work out the genetic diversity and what's going to be healthy, and we can start to rejoin those populations in a safe and controlled manner. So these are groups of animals that should have been connected, but through habitat destruction and roads and other issues, they can no longer meet to mate and to share that genetic diversity. Yeah. And so that's where people and conservation breeding programs can come in to actually introduce those animals, ensure they find each other attractive, that there's mate choice, ensure they have healthy offspring, and those offspring can also breed. And then you can increase the genetic diversity without swamping those local populations and get them back out safe in the wild in a healthier way and with better adaptation and survival for the future.
And we'll come to this, but you're doing incredible work breeding different populations to come up with more robust population. But why you're so hopeful is that you were involved in a project that had the most extraordinary outcome. So just give us a glimpse into the extraordinary story of the eastern barred bandicoot. 30 years ago, what was the reality it was facing?
The mainland eastern barred bandicoot is a
The
beautiful
cutest
story
little
of
creature.
hope. They're beautiful. They actually live in the grasslands like the grassland earless dragons in this unbelievably endangered ecosystem in Victoria. But 30 years ago, they were going extinct in the wild. And the last group of those animals in 1989 was brought into a captive breeding program to save the species. And it's a program that Zoos Victoria led for 33 years. And it involved a number of very important steps. There was appropriate focus and funding and partnerships.
Money was thrown at it.
A lot of money, a lot of passionate people, a lot of amazing partners to continue that captive program, to have animals released into safe, fox-free, fenced areas, because foxes were the major issue for these animals, and to recently put them out onto islands as well, like Phillip and French Island, where their numbers can expand. And in 2021, the mainland eastern barred bandicoot was taken off the extinct in the wild list and back to endangered because their numbers were looking so healthy. And our 33-year captive breeding program, just down the road at Melbourne Zoo and at our zoo properties, could be shut down because we were no longer needed, which is a world first.
A world first.
For a species listed as extinct in the wild to have a captive program closed, it's a world first. And it was an extremely important part of my career because at that point I was very privileged to be leading that program to its completion. And so the first step was stopping them from going extinct. The second was building up those numbers and working with partners and researchers and finding out what we could. But now genetic rescue comes into it. Because while we've had this success and the animals are doing well, at Melbourne Zoo, we had 23 founders. So every mainland eastern barred bandicoot can be traced back to those 23 founders at Melbourne Zoo.
Really? So all the eastern barred bandicoots that are around, out in the wild, are associated with that program?
That's right. They all
Wow.
have founders
God.
on the mainland.
That says to you, though, how vulnerable they still are. They're still endangered.
They are still endangered. We have them in multiple areas now and their numbers are growing. But through the Bandicoot Recovery Team and through Melbourne Uni, there's a genetic rescue underway to bring in new genes from a very similar and very related Tasmanian subspecies. And that breeding work's been done in a controlled wild area behind a fence at Mount Rothwell, and it's been successful. So those genetically healthy animals are now being moved across the populations to give them a boost and have them as healthy and successful as possible in the future.
I will come back to the question of how hard it is to do captive breeding, because it's not a straightforward thing, and you probably have lots of grief along the way and, you know, little populations that don't make it, and let's come back to that. And Muigai, Kenya. So what fantastic beasts are you charged with rescuing as a geneticist? You're not looking at Africa's multitude of wild animals, amongst the most diverse populations on the planet. You're looking at something much more domesticated.
Yes, and there are groups, I must acknowledge, who are looking at, you know, the big five, the elephants, the cheetahs, the rhinos, and I know you have been involved in some of those programs, and that's going very, very well. But not many people realize that the African livestock, the farm animals, the cows, the sheep, the goats are also being threatened. And when you think about African livestock, they've been on the continent for thousands of years. I think they're dated up to maybe between 20,000 and 12,000 years, some of them a bit longer, which means they're completely indigenous to Africa. And they all didn't originate there. They must have been brought in by the very, very early pastoralists. But these livestock are completely different from all the non-African species that you have out there.
How are they different?
They're different in the sense that they have a very, very large genetic base. They came at a time when they had to adapt to this new environment very, very quickly. It was marshy, there were lots of diseases, they had to walk thousands of kilometers. You can imagine from Egypt, we have livestock that walked all the way down to Cape Town.
Wow.
So over the years, over the centuries, and through that they developed adaptations. So some of them have very long legs and they're very agile in the way they walk. Like the livestock found in Central Africa, where it's very, very hot, have lost their color and therefore they're white to be able to deflect sunlight. And some of them have very, very big horns that are hollow, and this allows them to swim across lakes. So
They're like
because they
floaties.
float,
Right,
yeah, they
I
don't.
love it.
They just have to, get into the water and kick a little bit, and they'll be able to swim across. So they're very, very different and very unique, and they're under threat because the pastoralists have, if you have to live and walk with this animal, it's part of your existence because it's giving you milk, it's giving you meat, it's Africans drink blood, so it's giving you some form of protein, right?
Yeah.
So you needed to survive, and what has evolved is an integrated culture between man and beast. So the animal became an integral part of the culture, an integral part of the way of life. So you find livestock are integrated into ceremonies. If there's a marriage, there's an exchange of animals between the two families. If there's a girl that's born, there's a certain animal that's gifted. Then you know, oh, that family has gotten a girl because the goat is brown. If it was black, that would have been a boy. So things like that, traditions that could only be fulfilled. There's an old professor of mine who's keeping a bull, and he says, this is the bull that will be slaughtered at my funeral. And I said, what if the bull dies? Oh, I have to refresh it every couple of years because it has to outlive me. Yeah? So you see, so those are the livestock we're losing. And why are we losing them? Because the exotic livestock that come from Europe mainly are very, very large. They're very attractive, and they produce a lot of milk. They
And a lot of
have
meat.
a lot of meat.
So they're attractive, and they have been imported in a lot, haven't they?
Yes, they came in with the first European farmers who brought them in as they were coming to settle into Kenya and East Africa, and the local farmers around, of course, saw these animals that were huge and not long-legged like theirs, and they were attractive, and so crossbreeding has happened over the years, and over the last 50 years or so, there's been so much crossbreeding that we're now beginning to lose these very, very unique livestock. So that's where we have come in to encourage the farmers, because it has to be conservation on the farm. We cannot afford to take this. We can possibly take them out and keep them in a farm, a government farm.
So no zoos,
But
doing captive breeding.
zoos are foreign to Africans. We've lived with these animals, so keeping them locked in a house is very, very strange for us because they've been part of our lives. We take them out to graze. It's a way of life. And then we come back with them in the evening. So the easiest and the most cost-effective way of maintaining these animals is actually in the farms with the farmers. So we have to encourage and sort of show the farmers the value and the benefit of having these livestock, and then listen to them to see what they want. They want more milk, they want more meat, and that's where the science comes in, because we have to use genetics to identify, the good breeds, the good markers, crossbreed these animals to be able to, encourage this farmer to keep this animal. Otherwise, they'll, sell them off and bring in the animals that we don't like. And when we do that, the farmers are very happy because then we've addressed their needs, and then they have an incentive to keep the animal with themselves.
So just talk me through then, what's your role as a geneticist? How are you working with communities? Like if you walk on into a community as a geneticist, a scientist from a university in Kenya, and you've got your genetic tools, what's the feeling amongst farmers about genetics and genetically modifying their precious beasts?
Well, I think they wouldn't take the genetic modification because they don't want their animal to be changed. They don't want the animal to sort of like disappear because this is part of them. They see it as part of them, their identity. So using genetic engineering or such high science to knock out genes or to do technologies like that wouldn't be easily acceptable. However, breeding, which is natural mating and reproduction and things like that, that is acceptable because they also select their animals. You can go into a farm and realize that these farmers have actually selected their animals. They know what traits they want in their farm, their animals, and they would easily give away the ones that they don't want, because you can go in and find uniform animals from a farmer who's never been to a genetics or a breeding class, and they've done this over the centuries. So this is a knowledge that's passed down from generation to generation. So you can't walk in there as, the geneticist from the big university.
In your white lab coat.
Nobody will listen to you because he will challenge your science, because he's been actively breeding on the ground. So you have to listen to the farmer, and we've modified our breeding programs to be community breeding programs. So what would work in the first world, if you would call it that, wouldn't work there, because we have to be able to breed the way the culture, it involves the culture and is acceptable. And that way you get the science integrated. You'll say, well, if you want more milk, would you like to consider this, and that sort of a thing. Because at the end of the day, you don't want your animals to look different, and yet they're improved because they will be slaughtered the next day.
So you're going in though and sequencing the genome of the indigenous cows.
We are. We're using state-of-the-art technology to get to know what is there. We are getting to know which animals are, disease-resistant, because again, they will tell you that these flies came and my animal got sick and died, and immediately it's a SESE area, therefore you need resistant genes, and you'll sequence and get to know which animals are resistant. But then you don't want to lose the look, the phenotype, the color of the coat or the size of the horn, because that is the culture of the animal. So it's integrating those two, the knowledge and the science, to actually address the needs of the farmer.
Well, it's also valuing and taking into account cultural, spiritual, social values.
Indeed. You have to.
Are there any frictions there?
I think we made our mistakes in the beginning, because if many times you go into a farm and they will tell you, why are you back here again? And you would have difficulties explaining, I've never been here before. It's like, no, you came and you drew blood and you went away and now you're back for more, So we've made our mistakes, and now we are very, very careful that whatever successes or results that we get, whatever publications we are producing, we are able to take that information back and to explain to them how they contributed to the science and where the science is going and how it's benefiting them. Because without them, they will never be able to, change their practices or improve them or integrate them into new practices, yes.
So what's the Africa Biogenome Project, this massive endeavour that you're the chairperson of?
So the African Biogenome Project is a project that started in June 2021, although conversations had been happening way before. And it's a realization that there was an Earth Biogenome Project that was going to sequence all the world's plants and animal species.
All of them?
All of them. They've listed 1.5 billion.
That's a project.
Yes. Nice project. It's an Earth And the question is, what about Africa? Are we going to wait for the Earth Biogenome Project to finalize their work in 10 to 15 years down the road and then say, now turn to Africa and let's now see what we can do there? So as African geneticists, we had the conversation, what can we do? And there's always a question of, do we have the capacity? And we realized, yes, we have enough genomics and geneticists spread around the world and living and working in Africa. Do we have the knowledge? Yes, many of us were trained outside Africa. Therefore, we had the knowledge and the capacity. Do we have the infrastructure?
But the issue is
yes and no.
So few African animals have had their genomes sequenced,
yes.
Plants and animals. And of those that have been sequenced, they're often done by institutions outside of Africa. So you don't get to keep that knowledge or drive that knowledge.
So Africans, and myself included, have been used mainly to provide samples. So we'd get a collaborator who's interested in a species. I've provided sheep blood to collaborators because that was the only way we could do the science, maybe one of the ways. But you see, what happens is that when the samples leave, then you also give away, access to the
knowledge.
Results, the knowledge.
The kudos, the scientific papers.
Yeah, the paper comes out.
Scientific funding.
You might be acknowledged, you might not, or you might appear somewhere, in the middle of a couple of people. And yet sample collection is the key to the project beginning. And so also to address that, the fact that for as long as this was happening, we're also having brain drain, where the only place we could be, capacitated was outside Africa. which meant that we had young early career scientists leaving Africa, getting trained in genomics or genetics outside the continent, and not being able to go back. So they're scientific exiles because they don't have a place to get a job or live or work to their satisfaction because the infrastructure
So you
doesn't-
lose them for good.
Yes.
Why would you want to what's the power of sequencing 100,000 genomes in Africa?
You see, in a snapshot, you get to know what the DNA has. We've just heard that DNA is a blueprint of life, which means if you want to improve your crops, your answer is there. If you want to make them drought resistant, now climate change is happening, they're plants that are living in the desert, which means they have genes for resisting the dry climate. So you just need to find those genes and be able to identify where they are and create maps to form linkages with crops and see, do we have this gene in existence? If it's there, can we enhance it? And that's the power of genetics, because you'll be able to do what plant breeders or animal breeders did over a long time, in a very, very short time. But you don't actually need to get rid of the plant breeders, because you need them now to be able to actually do the work. And that's if you want to conserve something in an environment that has changed, you need this plant or animal to adapt to that new environment. So you needed to, it was used to living in a marshy area, now the soil is dry. Your answer is in genetics.
You've got to know what's there. You've got to document what's there in order to understand how to utilise it.
Exactly.
And enable it to thrive. Joanna Sumner here at Museums Victoria. So you're in charge of the DNA here in the collection. So just tell us about how the power of a museum to be involved in genetic rescue, because I guess you've got a building full of fur, skin, carcasses, pickled animals in bottles that have been collected here over, centuries now.
Yes.
What's the power of that collection in the genetic rescue story?
The beauty of a museum is that it's a collection over time and space. So by looking back in time at the samples that are in the collection, we can have a look at change. We can document the change that has happened over time. So we have a number of projects where we can look at the genetic diversity that is held in skin from...
Maybe an example.
So like some of the native mice so the smoky mice that we've...
So cute.
Very, very cute. And were thought to be extinct. But going back and having a look at some of the skins collected from this species in the last century, we're able to have a look and see how genetically diverse it was across the Australian continent, but also identified that what we thought was a similar species on one of the islands off Western Australia was actually the same species. So what we thought was an extinct species, we now, using the DNA sequences that we could get out of museum skins, we were able to identify that it was still alive, but there'd been this massive loss of diversity across the entire continent. So this collapse of a species.
And now they're on an island, which means that it's a concentrated genetic population, isn't it?
Yes, and that it's something where there's been a lot of loss of genetic diversity, and it will require ongoing management to hopefully try and increase any diversity that's there.
What's really exciting is that now you're not just keeping dead animals and documenting the loss of species, but you are also building a biobank of living cells. Tell us about that and why that's so potent in this story.
Yeah, so this is sort of another way we hope that we can invest in conservation. So by having a look at the genetic diversity in the biobank, in samples that are in the biobank, we can document loss of genetic diversity, but we can't actually put it back. So those cells that we have from just tissue samples that are frozen, we can look at the DNA, but the cells are no longer living. The current program that we've just started, the way we grow and then preserve these cells and the way we freeze them down very slowly means the cells themselves are still alive, and when we thaw them out again, quite slowly, those cells are living and able to divide. And then there's all sorts of possibilities for what we do with those cells. So we can, if we've got most of the cells that we're growing are fibroblast cells, so they're from skin cells. So if we take an ear snip, like
which you did with the grassland earless dragon, didn't you?
well, that was a tail tip we took.
Tail
so
tip.
with these very
important.
precious, earless has no tail. I mean, no ear
no,
snips.
it's
in
got
those
no
ones.
ear. That's right. Whoops.
And that's not why they're earless either. So
that's what the scientist tells us. Yeah. Okay, so you took a tail tip.
We took a tail tip. So of course, with only 16 individuals, we don't want to do any damage to these animals.
No.
We want to minimise any damage. So we took probably 2 or 3 millimetres of the tail tip of 2 individuals, one male and one female, and we then grew these tail tips in a specific medium. We added some other chemicals to stop them growing, bacteria and fungus.
So you've got these living cells.
We've got these living cells.
What's the promise of those living cells? Are we talking about one day creating a clone just from that cell? So inserting that, taking the nuclear material, the DNA out of that cell, popping it into the interior of an egg, and then breeding, cloning?
Something like that. So probably an easier option is something called an IPSI cell. So these are induced pluripotent stem cells. So they are regular cells that have been turned back into the kind of cell that can grow into any other cell. So like in an embryo, the original cells can develop into lung cells or heart cells or muscle cells, and those are pluripotent. They can become anything. So there's a potential for us to take these fibroblasts, turn them back into these IPSI cells, and then there's a potential to turn them into gametes. And that's where there's potential to get these living cells back into wild populations. If we have a gamete, if we have a sperm or an egg, then there's potential to start get them into the breeding programs. But there's also really other cool things you can do with these cells that you grow. So with this particular lot of cells, I extracted the DNA from it, and I had enough DNA from that we grew from that 2 millimetres of tail tip to sequence the entire genome of this animal. So normally we wouldn't be able to do that with 2 millimetres of tail tip, but having these live cells, we are able to multiply enough to sequence the whole genome.
Amazing,
so
Joanna,
cool.
amazing. Gosh, let's come back to crossbreeding, and I want to come to your questions now. So thank you for pouring them into the Slido platform. And it's timely that one of you has asked, does crossbreeding 2 species not fundamentally change the species itself? Marissa Parrott, now, so you're involved in crossbreeding projects with a number of different species, the helmeted honeyeater bird, there's a snowy mountain skink, I think, that you're involved with crossbreeding.
Skink,
the
yes.
Guthica skink, I think that's the mountain pygmy possum as well. Right. Some really interesting projects. Just tell us about how that's working and answer that question for us.
Yes, very happy to answer that question. And what we're doing is actually working with different populations of the same species. So it's not actually crossbreeding different species that might cause all sorts of problems. They certainly, for many of them, wouldn't be able to breed and produce fertile young themselves. But for animals like the mountain pygmy possum, there are different populations of that same species that have been separated for about 10,000 years. So they do have different genetics, but they can still produce young and produce fertile young. And so what we did there...
if you meet those 2 different populations.
That's right. So at Healesville Sanctuary, we did the first trials to show that genetic rescue was possible and that you could mate these animals. They were happy to do so. They produced fertile offspring. And then the mountain pygmy possum recovery team and the Uni of Melbourne went and did it out in the wild to help protect the populations at Mount Buller. For the helmeted honeyeater, we're working with amazing professors and teams at Monash University, so that's Professor Paul Sunnox and his team, and that's looking at a very closely related subspecies, which is the tufted honeyeater. And again, it's breeding these animals that actually do occur in the wild together and do breed in the wild together.
They do, right.
They do.
Because that's key, isn't it?
It is. Yeah, so it's really working with these animals that are so closely related that they can produce fertile offspring, but you can give them a genetic boost. And with the Guthaga skink, again, a beautiful alpine species, at Hillsville Sanctuary, we worked with 2 populations that have been isolated from each other, but in the past would have been continuous. They would have naturally bred if not for development and changes to climate and roads and so on. And so it's working with experts in the zoos, with experts in the wild, with amazing reptile experts who can see where these animals would have been joined. We can breed them and show it's possible in the captive breeding programs, and then it can be looked at out in the wild to see what's possible for the future. And so it really is just helping nature do what should have happened anyway, rather than breeding very different species that would have a very different outcome. We're just trying to do what should have happened in the first place.
Joanna?
And wasn't with the Guthaga skinks, it had a big effect on the number of offspring that were being bred in captivity? Wasn't there a big increase following the breeding between the 2 populations, from memory?
With many of the species there is, because you are increasing that genetic diversity, and you don't want to swamp the local populations and that genetics that has evolved for that particular area. You just want to give them a boost so that you have increased breeding success, as you said, increased health and resilience for the future. And so it's really important to do these kind of things in a conservation breeding program first, in many cases, just to make sure that there aren't any issues, that you don't get an outbreeding depression while you're trying to fix an inbreeding depression. And so that's just making sure there aren't any of those alleles that don't quite match up. But in all of these
alleles
cases...
being a variation of a gene.
Yes, so that genetic diversity that might be too different or that might be a problem. But in all of these cases, it's been really successful, and we've had beautiful offspring born and been able to then contribute to helping those wild populations.
It's not always successful though, is it though? You try and captive breed and they don't always survive.
Oh, if captive breeding were easy, anyone could do it. It's not. And that's why there are people who are specialists and reproductive biologists and geneticists, because by the time these beautiful animals come to places like zoos and sanctuaries, they're often already endangered and critically endangered. We don't know a huge amount about their biology or their diet or what they need. And so we need to work out their biology. We need to maintain natural behaviours. We need to work with geneticists and museums to look at what their genetic diversity is like and what they need to breed and to survive. And we're already pushing a boulder up a hill there because we've come in at this late stage where a lot of genetic diversity has already been lost.
So you've really got to get to know what they like and love, and that must have some challenges. Have there been surprises? I think someone here has asked, it was mentioned that smoky mice smell nice. What do they smell like?
Oh, they do. They smell lovely. In fact, it's one of the species that is on our list at Zoos Victoria as well. We work with Museums Victoria on the smoky mice, and they smell like vanilla. And so it's really quite lovely. They don't have that traditional mousy smell. Our native mice, our native rodents actually smell really quite lovely. And so vanilla, some of our native rodents smell a bit like walnuts. They really are a delightful animal in every way, and they're worth saving, and not just because they smell wonderful.
There's so many great questions coming in and I want to get to a whole lot of them. But a question for you all, does care need to be taken when reintroducing a wild species to avoid unbalancing the existing food chain? You touched on that, but also how can we make our urban environments, or any environment I think, more hospitable to endangered species that are being released from breeding programs? I'm interested in all your thoughts about this, because this process of kind of not engineering, but kind of curating a crossbreeding process only to kind of reintroduce that species back into an environment where it's already been compromised. That's an issue, isn't it? This is human hubris at work, potentially. Any thoughts?
I can jump in with that one to start
Marissa
with.
and then Anne.
So when you're working with an endangered species, it should be out in the wild. And the main focus of our work is to actually get species out in the wild, safe where they belong. But you also need to make sure they're released into a safe location. So with the eastern barred bandicoots, it was into fox-free fenced areas and islands. With a lot of the animals where we're doing genetic rescue, they may still be out there in the wild, but in those small populations. So we're putting new animals in to give them a genetic boost, but keeping them out there safe in that ecosystem and working with partners to control predators and to do revegetation. You want to make sure that those animals go out into safe locations where they can thrive. And people don't realise that while that is often a long way out, whether it's in the Alps or down in East Gippsland for tiny little native rodents like Pukila, we also have a huge number of endangered species around us. Here at Melbourne Museum, in the skies above us, you'll find bogong moths flying at certain times, which are endangered. You have critically endangered swift parrots and endangered gang cockatoos. And there are ways that we can protect these animals by doing simple things, like turning off outdoor lights so that the bogong moths can continue their migration, and tracking them on our citizen science website, Moth Tracker. You can plant native plants to help support the gang cockatoos that come down to feed. And importantly, you can keep your pet cats, which we love very much, contained inside so that you have a safe cat plus safe wildlife. And that way we can all make a difference to the animals around us and those that are further out in the wild as well.
And Alejandra has asked, what are the biggest risks with genetic rescue? From your perspective, what are the biggest risks of genetic rescue?
Well, some of them, you make assumptions when you're going in to rescue a species, and you might have got it wrong in the beginning. We've just heard about how biology is so important, and that's why you can't go into a genetic rescue project by yourself. You have to bring in the ecologists, the environmentalists, and all along the food chain. For instance, you might be targeting to release an animal into the wild, and case in mind comes the African rhino, which is critically endangered. I mean, this is an animal that is under threat, so you can't just release it. It has to be safe. And safe for us in Kenya means that for every rhino that walks free, it has a guard. It has a armed guard following it around, because, I mean, we're down to the last 300 or 400, and we can't lose even one. So you have, sometimes you can decide, I'm going to take it to this environment, but you've mapped it with old data and the environment has changed. And maybe there's no food, or the grass is too dry, or whatever it is. So you have to be able to bring in a team of people who will, at least now with science, you can model the environment and do some modeling experiments in a computer before you actually do some of the releases, and that will safeguard the safety. Remember, you've spent so much time, so much money, so much resources understanding this animal. You can't afford to lose even one. You want it to go into the wild and to be able to reproduce, and that's when you know that you've been very successful.
You're working with meat-producing animals and trying to protect their diversity, the indigenous meat-producing animals. And Ben has asked, and I'm interested in your response to this, Ben asks, our global appetite for meat as a staple food places so much pressure on endangered species. What do you see as the change that gets us closer to sustainability? I mean, that must surely be an issue for you. If land clearing is threatening wild species in Africa, that land clearing is partly to accommodate the domestic species that are being used for livestock.
And also remember that as people move towards the cities, the lifestyles change and the demand for meat also grows. So just change, sort of like progression and move. This urbanization has also created different tastes among the people, and therefore we've become more hungry for food, for meat rather, and that has created a big demand. And therefore the balance, are we able to use indigenous livestock to be able to meet this demand? And if you say no, then the farmer will not benefit from the sale of his animal. If you say yes, then are we able to reproduce and replace those sales as far. So it's always about creating a balance. And therefore, there has to be another model that's also helping us supplement that. There are always the large-scale farmers who have the investment and the ability to do the exotic animals, and that's also feeding into the food chain, and we need that. And we also have the crossbreds, which have the best of both worlds, the high genetics of the exotic animal, plus the traits and the resilience of the indigenous, and that's also helping us to bridge the gap. But you can't go it in any way, and we're still meat deficient, and some African countries are having to import meat from the rest of the world.
Yeah. Lella, who's 10, where's Lella? Maybe you're online or you're in the audience, but great to have your question. Can you revive extinct species, for example, the woolly mammoth, and should we? Joanna Sumner.
That is a very tricky one, the should we. I think, again, it's going to depend on the situation and which species you're talking about. So there are species that we're trying to bring back from extinction.
The thylacine.
So
and someone else has asked, how do you feel about recent efforts to bring back the thylacine? Now, the museum's somewhat involved with that.
We are. So one of our collaborators, or we are collaborating with the thylacine group, and I think, so I was initially unsure about what they were trying to do, but I think it's going to be the important things that will come out of the work that they're doing in trying to bring back the thylacine is all the knowledge that we will gain on the closely related species that they're looking into. So they're talking about editing some of the genes within a dunnart, and which is a little marsupial, which they're very ferocious, so just by doing some of the changes, but also the research that goes into this species will have really important conservation outcomes for that whole group. So overall, I think the project has a lot of benefits. I don't know whether they're going to get a sort of slightly more thylacine-like dunnart out the other end, and it will depend on the research that comes out and the successes that they have. But I do think that project will have really positive outcomes. But that doesn't answer the question of whether we should bring them, should we? And I think we should try. I think for things that we are recently responsible for the extinction of, and that there's still habitat available, like the thylacine. There's beautiful Tasmanian habitat available, as Marissa was talking about in our previous conversation before we started.
But all the animals in Tassie are also struggling to exist. There's a lot of roadkill in Tassie, those beautiful wombats and their mange, the cancer facial tumour.
Yeah, but it may be that the loss of the thylacine has put the whole ecosystem out of whack anyway. And bringing that top predator back into the system may have the sort of follow-on, knock-on effects that bring the system back into some more similar to what it was before the loss of those animals. So I think that there may be space to do it.
So here's a question then that's related to this, and how are species prioritised for rescue within individual countries and continents and globally? Marissa and Anne, do you have a thought about that?
So thinking about prioritization, we've also been grappling with that question in the African Biogenome Project, because which species do you begin to sequence first? How do you prioritize? Well, we had a lot of submissions from different parts of the continent, and we came down to the one that is going to address a need that is, really, really pertinent at that point, be it food, it maybe is a crop that was indigenous. We're also looking at the indigenous species that are only found in Africa, species that are found in what we're calling biodiversity hotspots. These are ranges with the largest numbers of species in a very short distance, which means that any destruction of that, even a mile of,
so much will be
lost. It means that you're killing so many or making so many extinct. It could be that it has cultural value, and I've mentioned some of the species that have great cultural value. So you have to think about the balancing, and it's not easy. But sometimes, always, the ones that get on the list first are the ones that are at the greatest threat, whether it's going to be whatever it is, a plant or an animal. The one that is going to disappear in our lifetime gets priority over everything else.
And it's often the charismatic animals that get attention too. And someone has asked here, wouldn't it not be more vital to place focus on critically endangered species rather than revive an already extinct species? For example, the Thylacine project. Marissa Parrott.
Yes, I can talk to both of those questions at the same time and absolutely agree with Anne that you're looking for species that are the most critically endangered. And at Zoos Victoria, we have that prioritisation process where we look for animals that have a declining population, a restricted and fragmented distribution, key threats, whether it's habitat destruction, introduced predators like cats and foxes, disease like the amphibian chytrid fungus, and which species could be at risk of extinction in the next 10 or 20 years if we don't help and our partners don't help. And so you're really going for those species that are really coming to the brink of extinction to stop them from falling over that cliff and bring them back. And it would be wonderful if we could look at the other species as well that are threatened, that are vulnerable, to stop them from getting to that point. But with the funding that we have and the number of people that we're able to have, we have to really focus on those extremely critically endangered animals. And so it's wonderful to see funding go into saving those critically endangered and endangered species. It's different funding that goes to things like the Thylacine project. And so there isn't really that competition. But as Joanna was saying before, the information we learn from the Thylacine project is around the genetics of related marsupials like the fat-tailed dunnart and the eastern quoll and the northern quoll, which are all threatened species. It tells us about assisted reproductive technologies that in the future may be used to bring back lost genetics that has been banked down in a biobank. And so really
lots of flow-on possibilities for other species.
There are many flow-on possibilities in the research that's being done. But for us and at Zoos Victoria and zoos and communities around the world, it really is stopping species from going extinct.
So I just want to squeeze in a couple more questions, but I'm going to collate. There's a number of questions that you're asking about numbers, if you're online or in the audience, and I'm just going to throw them all into one question. How big would an ideal population of a vertebrate be in order to be secure into the future? What is the minimum number of individuals? That's from Kate. Anonymous asks, if 50 years wasn't long enough, when does a species get declared extinct, or what are the criteria? And someone else has asked, how many individuals were used for the founding population at Zoos Victoria? How will the zoo ensure that the earless dragon does not have inbreeding? Well, that's a sort of slightly tangential question, but numbers. Interesting. Any thoughts on those, just really briefly?
Very briefly, it's species specific, and so it depends on the genetics of the animal, on the generation length, how long they live. It depends on the areas you have in the wild, and so you really can't put a set number for many of the species we work with. For that particular species, say eastern barred bandicoots, we want to have at least 2,500. For Pukila, we want to have at least 5,000. For animals like the bawbaw frogs, their numbers are extremely low, and so we're looking for increasing those numbers as best we can. You know, it really is different for all of those different species, and there are criteria under the IUCN, the International Union for the Conservation of Nature, which tells you what kind of aims you need for certain species. It talks about why different animals are in different criterias based on the numbers, based on their decline, based on things like their geographic spread. And so there are so many different aspects that go into working out what your aim is. For many of the animals we're working with, the numbers are in the hundreds, as Anne was talking about before. For our grassland earless dragons, we're starting with 16. And
only 4 are female.
And only 4 of those 16 are female, and that's why it's so important that we're working with...
pressure's on.
It is.
Yeah.
It is indeed.
They have an amazing team there, though.
We do.
At the zoo. Yeah.
We know we can breed them because we've done the work already with a similar species, and we know that we can start to increase those numbers and look for more with eDNA and detection dogs. We're not giving up on any species, no matter how small they get. and we're going to do everything we can to bring them back.
So I want to wrap with a question that lots of people have voted for me to ask, and I'm sorry, I've been jotting around all the questions in different ways, but advice for young women in wildlife or science getting started in a career. Joanna.
I think probably volunteer. I think volunteers are fabulous and they really keep a lot of projects going, and it gives you an opportunity to see whether you actually really like the work that you're doing, and you can test out a few different things. So if you have any opportunity, I know you should all be paid for all your amazing work, but it just may not be possible. So that would, see if you can volunteer.
I'd add and say that get a good broad-based biology first degree that will give you insights into plants, animals, and ecology, and from there you'd be able to identify what you'd really like to do, and you can move on from there.
And I'd agree with both of those, and both of those getting that biological background, doing the volunteering, following your passions means that you're networking and you're building up your skills to make yourself more employable. And having those networks is extremely important when you're looking at what jobs and what projects are coming up, what skills can you offer others. But it really is following your passions. If you love something deeply, you'll be good at it, and that will show. And so just don't give up. There's always hope out there.
Well, speaking of hope, what keeps each of you hopeful? Because there are so many species, so little time. Humans are kind of the scourge of the earth, let's face it. And so what gives you hope? Because it is a field kind of riddled with despair as well, isn't it?
Absolutely. But I think I actually get hope in the science. I find hope in the science, and I find hope in all the things that we're discovering, and how I think the more we learn about the world around us and how extraordinary it is, I think, and if we can get that information out and more people find out about the extraordinary world and its complexity, I think the better. And so I think, for me, the science gives me
is hope.
Yeah.
And Victorian grassland earless dragon, that's a hopeful story.
Absolutely.
Anne?
Well, for me, it's going back into a community I worked in, maybe exited when they didn't need my support, and driving by or stopping by and finding my livestock thriving, the community thriving. They've got the genetics on point and they really don't need me anymore. And my species is upgoing, and I'm thinking, well, it's time to now use them to move into another community and see whether we can make a difference. And that just keeps me going. It's just so much fun to see that you're making a difference, farm by farm.
Yeah, farm by farm, community by community. Yeah. Marissa?
I think for me, it's seeing those wins that we have on the board already, knowing that we can save these species. We can find the grassland earless dragon that's been missing for 50 years. We can take the eastern barred bandicoot on mainland Australia off the extinct in the wild list. We have a new breeding program for beautiful little pukilas, a native rodent that's been described as a dumpling on legs. And we've bred them, and we're going to be preparing for releasing back out to the wild. So seeing those wins for these animals that are so endangered gives us hope for all of the other animals out there that together we can do it. We can save these species.
And for people who feel powerless, they don't have the tools of genetic science at hand, and they're not part of these projects that you're part of, what can give us all hope that we can help make a difference? I think that's a vital one. Someone has actually a very controversial question here. I have a dog in my life, and so I will ask this. Should pet cats be prohibited or banned in Australia? That's a radical question, isn't it? You don't have to answer it because you might get in trouble from all the cat owners. But what keeps us all hopeful? Because it's easy for us to feel a bit oblivious to the fantastic little beasts around us that we share this planet with.
I can jump in as a cat owner as well. So I have both a dog and a cat. My dog goes out for walks with me on lead and in controlled areas, and my cat is a very happy indoor little cat. And so I don't think we need to give up the animals that we love. We can have them safely inside with appropriate pet ownership. And the cat's out of the bag, to put it mildly. There are feral cats across Australia. So what we need to do is take care of our own backyards, work with different techniques and technologies to try and protect our wildlife and decrease the numbers of feral cats. But there are ways that we can keep cats happy inside and make sure they stay inside. And in fact, we have a Safe Cat, Safe Wildlife program where people can learn how to do that. So you can still have your much loved pet cat, but you can also make sure the wildlife is safe and you're not contributing to creating a bigger problem.
Absolutely. Anne?
I think for me it's the realization that we're there in the nick of time and we have a very big opportunity to make a difference now. And we shouldn't wait for others to come and, provide the money or do the science. We are there now, and therefore we can lend even our voices, if we're not in that area, to raise awareness. And we have the science now. We didn't have it in the 60s. There's all these technologies that are coming out that can be put to the good, and basically, let's act now.
And finally you, Joanna.
I was so busy listening to
What
what Anne was
citizens
saying.
what citizens feel empowered by?
I think for me a big part of it is gardening and growing native. Native
Yay!
garden.
Yes.
So
Plant for the birds and bees.
Yeah. So I have a beautiful native garden out the front of my house. I grow veggies out the back, but I often watch the native birds that come into my front yard, and really love that. And that's, I guess, one of my ways of doing something is adding some habitat.
Love it. Well, I've found this conversation extremely hopeful, and it just really gives you a sense of what's possible, doesn't it? But it also raises a whole lot of questions for me too about humans and how we feel we can solve the problems that we've created by actually potentially creating even more problems. So that's always a question too, isn't it? And thank you so much for your wonderful questions. And you have the opportunity to give feedback. I think there'll be a survey. We really want you to do survey. I think that it will be emailed to you. You go in the running for a $50 museum store voucher. So please do that, because it helps this wonderful institution, Museums Victoria, to build future events for you. Also, the next Future Forum is a very exciting one indeed, live from, I think, a campsite somewhere. David Suzuki is going to be joining us for the Now or Never Festival. The Now or Never Festival is Melbourne's brand new festival that brings together the Melbourne Innovation Festival and the Melbourne Music Festival. It's coming up very soon. That event is on Saturday the 19th of August. It's already on the Museums Victoria website. A live conversation and oration from the wonderful environmentalist, trailblazing environmentalist, conservationist, Canadian David Suzuki. So get on and register to be part of that event, and I think it'll be streaming live as well. We were recording today's event for Big Ideas. You can follow Big Ideas on the ABC Listen app. Just download it to your phone and you'll find me there every night of the week on the ABC. Please, let's thank our wonderful speakers. Anne Muigai from Kenya, thank you so much for being here at the Genetics Congress. Marissa Parrott from Zoos Victoria, and Joanna Sumner from Museums Victoria. Enjoy your night. Thanks for being here. And if you're at the Congress, have a great week. I hope it continues to be a fantastic Congress for you all. Thanks so much.
The Museums Victoria Research Institute
is reimagining research,
drawing on First Peoples and other knowledge systems
to expand our understanding
of our rapidly changing world.
We're seeking your support to help make an impact,
conserving Victoria's most vulnerable native animals using cutting-edge cryopreservation technology,
working with regional communities to lead innovative citizen science projects,
to proactively connect the First Peoples' cultural inheritance, which is in our care, with the people and country it belongs to. Donate today and make your impact. Please.